All right, good afternoon, everyone. Welcome to this special seminar this afternoon associated with a hire in ASU Center for Evolutionary Medicine and Public Health. So it's a pleasure to introduce this afternoon's seminar speaker, Andrew Reed, who's with us from Penn State University this afternoon. So to give you the particulars, Dr. Reed has his bachelor's degree from the University of Otago in New Zealand. He asked that I mention this specially so that you could identify his voice and where his accent is from without any problems. Went then to University of Oxford, has his doctorate from there after working with Paul Harvey at Oxford. From Oxford, he had several adjunct appointments, but then settled at the University of Edinburgh, where he was a professor of natural history, holding a chair established in 1767. I like the fact that he was a professor of natural history, since my professorship is in natural history also, which means there are two of us in the world with professorships in natural history in the 21st century. So from Edinburgh, he goes on to Penn State University, where he is now and in the Center for Infectious Disease Dynamics. He's also holds professorships in the Department of Biology and Entomology. Dr. Reed has several awards. He's a fellow of the American Academy of Microbiology, an elected member, a fellow of the American Association for the Advancement of Science. He's a fellow of the Institute for Advanced Studies in Berlin, fellow of the Royal Society of Edinburgh, and he also holds the Thomas Huxley Award from the Zoological Society of London. As I said, this is a special seminar connected with the Center for Evolutionary Medicine and Public Health. What we'll do is we'll have the regular seminar, there'll be time for questions, and then those of you who wish to stay, we'll have another hour for a Q&A with Dr. Reed for those who would like to learn more about his research and his interests relative to teaching. So now, let me turn it over to Andrew to talk to us about pathogen evolution in a vaccinated world. Andrew?
Thanks, Fred. Thanks very much, Jim, and I'd also like to say thanks to Randy and everybody else who's done a great job on organising the trip so far. It's been extremely stimulating, and it's lovely to be in the sun away from the north. My group works on my group works on the evolution that happens in disease-causing organisms when we do things to them directly or to the world they live in. And much of that is tied up with what we do when we wage chemical warfare through drugs or vaccines or insecticides against the organisms that cause harm. Now, I talked here a few months ago about the work we've been doing on drug resistance, which is about half the work we do. And so I'm not going to talk about that again. I just want to give one little nugget of that work. You all know that when you go get antibiotics, the doctor asks you to make sure you finish the course of antibiotics even after you feel better. And there's usually stickers and advice and stamps. And our work on drug resistance has suggested that that's not a very that's not a well-based piece of advice in evolutionary theory or in practice. And next time you get antibiotics where the doctor says, please take them for longer than you need to, ask them why that it's worth continuing to impose selection for resistance long after you don't need the drug. I don't have time to talk about that anymore. Where the drug resistance work is going, though, is I've got a research leave at the moment at the University of Michigan in the hospital there, in the Department of Infectious Disease, and what I'm interested there is the evolution of drug resistance in real time in the wealthiest country in the world. People die in this hospital, and indeed your hospitals, from resistant infections which are untreatable. And that evolution is happening in real time, and so I'm very interested in what we can do about trying to slow that down. The aim of the game of all of this work is to try to slow down that evolution, or to try to avoid mismanaging the evolution and making the situation worse. And the sort of problem that's coming up in the hospital, which I've been really startled by, is, for example, we have a patient who has a chronic infection that can't be cleared. And she has 2 drugs left. So the infection is now resistant to everything else, bacterial infection, resistant to everything else now. And Bob Woods, who I'm working with, said to me, let me introduce you to this patient case. And then when he finished up summarizing that, he said, there are 2 drugs left. What are we going to do? My first reaction is, what do you mean, we? But the question is, how do you use those drugs? Successively, together? Do you rotate them? Do you give her a break with no drugs at all? Do you combine it with some of the drugs that have been used before but failed? What do you do? How do you treat that patient in a way which slows or stops the evolution of resistance which is going to kill her? I have some theory on that and some ideas on that, but we're a long way off coming up with a solution to that particular patient problem or related problems, including the ones that I've just been talking with people in the Mayo Clinic here this morning about. This real-time evolution is really important. So I'd love to talk about drugs, but as I say, I did do. So I'm going to talk about our work on vaccines, and that includes some of the work that we've been doing on farms. And what I'm going to try to do in this talk is make 4 what I think are relatively straightforward points, which I'll step through as we go. The first question I want to address is the difference between drugs and vaccines from an evolutionary perspective. And this is a history of the rollout of anti-malarial drugs in the last century. So chloroquine went into widespread use at the end of the Second World War. Chloroquine resistance was first seen in the field 10 years later. So infections that no longer responded to treatment were first seen 10 years later. Proguanil resistance, pyrimethamine resistance, and so on. So this is one disease for which we have 9 or 10 drugs, all of which have failed. Artemisinin is failing at the moment, the latest drug. It's failing at the moment. And so what we have is a drug discovery treadmill where we need to invent new drugs to replace the ones that have failed because of pathogen evolution. This is true in antibiotics and so forth. Now consider the difference with that in the vaccine case. So in the vaccines, we've had a series of vaccines, some of them very old, that have been rolled out, and they haven't failed. They work really, really well. In fact, they work so well that they've managed to eradicate diseases. So these pathogens have not evolved around these vaccines. So now what we've got here is 9 or so diseases, each of which has an effective vaccine which has contained these pathogens, and there is no vaccine discovery pipeline necessary for these diseases. So there's quite a difference between what happens when we wage chemical warfare with our drugs and what happens when we wage immunowarfare with our vaccines. And that's led some folk to suggest that vaccines are in fact evolution-proof, that they're different from drugs, that the pathogens are very good at evolving around drugs, but they're not very good at evolving around vaccines, and so you don't need to worry about vaccine-driven evolution. And the argument, to the extent that anybody's ever given me one that's got a theoretical basis, is broadly speaking that immunity is happening anyway. The diseases induce immunity, and when we use vaccines, all we're doing is replacing disease-induced immunity with vaccine-induced immunity, so you haven't changed anything as far as the bug is concerned, and that's quite different from drugs, which they've never seen before. Now, I think this contention that vaccines are evolution-proof just can't be right in principle, and there are, broadly speaking, 4 reasons for that. The first one is, as many people in the room here know, because they're working on vaccines against hard diseases, is that there are a whole bunch of diseases which seem to be very naturally incapable of being vaccinated against, precisely because they can evolve so fast. Their flexibility allows them to get around vaccine-induced immunity, so we don't have vaccines, commercial vaccines against them. The second thing is that we often do see pathogen-driven evolution. And I don't want to go through the details of each of these, just to say that here's some cases where after the vaccination has been introduced, where the green arrow is, then non-vaccine alleles rise in the bug populations, or in the case of diphtheria, the toxigenic strains decreased. So you can see, you can measure evolution in real time in response to vaccination. And it's the case that some animal vaccines have failed in the face of pathogen evolution. I'm going to talk at quite some length shortly about a chicken vaccine that we've been working on, where that's happened. But there's also other examples. I'm going to briefly mention this feline calicivirus, which is, if you're a responsible cat owner, you've vaccinated your cat against this. there have been two separate evolutionary events where very hot strains broke out in high-density situations like catteries and tore through vaccinated cat populations. So strains popped up which could easily get through the vaccine, which is very, very good at protecting against the ancestral form. And though, in fact, those two evolutionary events, both cases, the outbreaks were so hot, they burned themselves out when they ran out of cats. They killed so many cats, they burned themselves out. And that's an example where the vaccination failed in the face of pathogen evolution. And again, as many people here are trying to do, many of our vaccines, we're trying to induce immunity in a way which is mechanisms of immunity which aren't the ones naturally induced by infections in nature. So that's going to impose novel selection. So I think these are the reasons why we should be suspicious of the claim that vaccinations is somehow different from drugs in a magic sense, and that we should expect pathogen-driven vaccine-driven pathogen evolution. But that leaves us with this problem of why is this history so good? Why has it worked so well? And I don't have a very strong answer to this, but I want to offer you an observation. This is a very famous data set on measles in England and Wales. I think there's at least 70 PhD theses have been done on this time series. You can see those spikes in the cases are pretty frightening numbers, and the vaccine era basically wiped things out. So the vaccination is very, very robust, and no mutants popped up here which were capable of getting through the vaccine-induced immunity. But just think what was happening at each of these spikes. At this point, the virus has run out of susceptible individuals in a population, and so it's crashing out. So at this point, there's immense selection for any mutants that could get into an immune person, a person who's seen measles before, and yet in each of these waves, that hasn't happened. So for some reason, and this is the bit we don't have a good handle on, for some reason, this pathogen, or the immunity that's generated by this pathogen, is naturally evolution-proof. This pathogen is incapable of evolving around the immunity that it generates. So it crashes out. And so I think if you look at that list of vaccines that have been very successful in the long run, they're all childhood infections. They specialize on fully susceptible individuals, and they elicit extremely strong sterilizing immunity, which is lifelong. And so that's why we call them childhood diseases, because they are stuck with children. They can't get into adults because the adults have already seen the infections. And so what's happening with these vaccines, I believe, is that they are simply mimicking a natural situation. They are mimicking evolution-proof natural immunity. Now, why that is evolution-proof, we could discuss afterwards, but that it is evolution-proof is, I think, pretty clear. These childhood diseases are childhood diseases because they have immunity which is not capable of being overcome by pathogen evolution. But that's in contrast to what I think of as the diseases that we are now vaccinating against or trying to develop vaccines for, and many of these are being worked on by people here. These are the hard diseases for which we haven't easily got vaccines, and these are the ones for which they are not childhood diseases by and large, and the natural immunity is not perfect. So under these situations, I think we will expect evolution to cause problems, and that the vaccines and the vaccine designers need to think hard about making sure that we can cope with the evolution that's prompted. So my first point is that the fantastic successes that we've got from these childhood vaccinations, and they really are fantastic, best healthcare outcomes we've had in medicine, except maybe with the hygiene, absolutely fantastic. That history is special, and it's special to special diseases. And the vaccines that we're trying to generate now and the diseases we are trying to halt now, these are very different diseases, and I don't think it's going to be anything like we've seen in the past for the 4 reasons I gave before. So that means we could ask the question, well, what will the evolution look like? If the pathogens are going to evolve in response to vaccination, what's going to happen? And that's what I want to spend the bulk of the rest of the time on. The standard thinking is that we will get the evolution of escape variants. And that's kind of like we think about in the flu case. The flu comes through, we get immunized by the natural infections, and then the following year the flu has to mutate a bit to get into the population again. And this is antigen AMA1 on Plasmodium vivax, which is an antigen that we've been using for vaccination. the idea would be that if you raise antibodies with a vaccine against AMA1, then there'll be mutations in the structure of that molecule which will mean that the antibodies don't bind so well, and those mutated variants will have a fitness advantage and they will rise in hosts and then in the population, and that will erode away the efficacy of the vaccine. And that process of escape and erosion is more or less the same as drug resistance. It basically means that if it went the full course and worked really well, that you'd have a vaccine that was very, very good to start with and then eroded away to nothing as the escape variants spread through the population. That's a very important form of evolution, and it's one that's considered by some people in the vaccine development community, and so depending on your disease it can be very important, but it is not what I want to talk about. I want to talk about the possibility that you could also get the evolution of virulence. That's to say, hotter strains of the pathogens. So when I'm using virulence is one of these words that's used in lots of different ways. When I use it, I mean the increased virulence means more harm to the host. So more virulent strains are those strains which cause more suffering and death. So the question I want to ask is whether or not vaccine-driven evolution could lead to the evolution of hotter strains, more virulent strains. And this, I got interested in this question following from our work that we've been doing on virulence in Plasmodium schabotii. This is a rodent model of malaria. We've been working since the late 90s with various different strains of this species. And this is data on two of the strains, the red and the blue. And this is the density of the parasites through time in mice, or the red cell density through time in mice. And this is two strains where you can see the red one is not growing up as high, doesn't get such high densities, it collapses down faster, and the blue one goes higher, and then it relapses more. So the blue one is more virulent, causes the mice greater sickness, and you can see that in the anemia curves. The mice are getting more anemic, fewer red cells with the blue strain than the red strain. And so there's strain variation in the species that we've been working with. Much of the first half of my career, I guess, is based on this strain variation and what it means. And this is a bunch of our clones and how much weight loss they induce in the mice. And so we go from a strain AS, which is relatively nice, through to ER, which generates another 2 grams or so of weight loss, which in a 20-gram mouse is a fairly significant amount of extra damage. And so we've been asking over the years what's the natural selection would be on that sort of variation. What would make AS dominate or ER dominate? And the upshot of a lot of that work is that what we discovered in terms of immunity was that the virulent strains are less well controlled by immunity, so they can persist for longer in the face of an immune response. And when we did vaccination work, that the virulent strains were better at breaking through vaccines. So that's what got me interested in this possibility that vaccine-driven evolution might result in more virulent strains. And we did a lot of work for many years along these lines until finally we got down and did a proper evolutionary experiment to ask this question of whether or not the parasites, malaria parasites, are capable of adapting in a genetic sense to vaccinated hosts, and if they are, whether they'll be more virulent. And here I mean virulent in the sense of doing more damage to an unimmunized host. As I say, we work with AMA1. This is a protein that's on the surface of the parasite, and it's involved in the invasion of the red cells. And you can elicit very potent antibodies that bind to that and prevent the parasite from invading the red cell and protect the host. So what we did, and the we here is the heroic Vicky Barclay, is the following experiment from hell, which I'm not sure we'll do too many of again. We took a single parasite, so a single malaria parasite, that's the one I've got up there, and from that clonally grew up lines. We've got what we call the C lines there, the control lines, and those are parasites that got passaged through mice that had never seen a vaccine or a parasite before. And then we've got the V lines that we set up, which we passaged through mice that have been immunized with AMA1, that's recombinant antigen, and so we can generate very strong, very potent antibody responses. And then we let the evolution proceed as we passage the parasites from mouse to mouse to mouse. Now we set that up with the aim of having 5 lines in each of those treatments. So you can see at the top up here, you see we've got the 5 lines we've set up here. And then each of these red dots is where the line died out. That's to say we didn't get enough parasites to passage into the next mouse. So what you can see is that we tried, we've got 5 lines going here, we lost 1 of them. We got 5 lines going here, we lost 4 of the 5 of the vaccinated ones. Okay, so you see just from the picture here that we had really no trouble keeping these lines going. When we lost a line, we just duplicated another line. You can see with the control lines, we really had no trouble keeping the parasites going. With the vaccinated lines, we had problems getting enough parasites to go from mouse to mouse, and that's because the vaccine is really good at suppressing parasite densities. You know, the vaccine works. And so you struggle to get enough parasites out to put into the next one. And so you can see we had carnage. We lost the lines, we lost the lines, we lost the lines, we kept losing lines. This used to happen on a Wednesday, and I hated Wednesdays because they'd come in and say, well, we've lost this many lines, what are we going to do now? But just look at the picture, you can see that after about 10 passages, we stopped losing the lines at that fearsome rate. So we started to get parasites where we had less and less problems passaging them on to the next host. So it looks just from this picture like the parasites are becoming better and better at growing in vaccinated hosts because we're able to get them into the next generation. If we actually go and measure the parasite densities in the mice, and so this is just putting all the individual mice together and all the lines, the blue dots are the parasite densities in the unvaccinated mice, and the red triangles are the parasite densities in the vaccinated mice. And you can see right at the start of the experiment that the parasite densities in the vaccinated mice are much lower than the parasite densities in the sham, and that's because the vaccination suppresses parasite densities. But over the 20 passages, the parasites got better and better at growing in the vaccinated hosts. And so the actual anti-parasite effects of the vaccination eroded away. And statistically, at least, after 20 generations, there's no difference. The vaccine is not killing parasites any faster at all than the sham after 20 generations of that evolution. So in other words, the parasites have adapted to this new environment and efficacy has eroded away. This is just to remind me to say that no evolution happened at the target locus. So the AMA1 locus in the parasites was identical. The sequence was identical at the beginning and the end. There was no variation. No evolution happened at the epitopes or the antigen or the gene or anything that we were targeting with this recombinant antigen. So the evolution did not happen in a strain-specific way. It did not happen in epitopes or antigens or any of that. And so what we did was to take the parasites at generation 10 or at generation 20 and take those parasites out, the evolved ones out, and put them into naive mice and ask, had the parasites got nastier? So were the parasites, the evolved parasites, doing more damage? And this is the results after the 10 passages and after 21 passages. So this is the parasites that have been evolved through the naive mice and through the vaccinated mice. And this is the ancestral parasite and through the naive and the vaccinated mice. What you can see is that the parasites that evolved through the immunized mice, they caused more anemia in the animals, they caused more anemia, than the animals, than the parasites that had evolved through the unvaccinated hosts. About 20-25% more anemia was induced by the parasites that evolved in the vaccinated hosts than those that evolved in the unvaccinated hosts. And so from the perspective of the evolution of virulence, I think what that within-host experiment shows is that pathogen-driven evolution can be about more than just the antigens, so it's not just the strain and escape variants involved, and that it's possible that under some circumstances vaccination can lead to the evolution of pathogens which do more harm to unvaccinated individuals. Now, in fact, there's another reason to think that vaccination under some circumstances could lead to hotter strains evolving. And that was actually the thing we got onto first. I'm telling this story slightly backwards in terms of the experiments we did. We got very interested in the early part of this century in the theoretical possibility that vaccination has this potential to generate hotter strains. And the argument went something like this. This is the sort of cartoon schematic. Ask yourself, why is it that more virulent strains aren't found anyway? Why are diseases, infectious diseases, not even nastier? And the usual explanation is that if the strains were really hot, they'd kill their host and therefore themselves, and they'd have no future. So from an evolutionary point of view, it doesn't pay to kill your host too fast. And so that is the standard argument for why we don't have super hot pathogens. And it is interesting that the ones that are really fatal, like a high case fatality rate like rabies, they're just not maintained in the human population. They're maintained in something else and spill over to us. Our diseases, our infections don't kill nearly at that rate. And the explanation for that from an evolutionary point of view is usually that killing your host is bad because you kill yourself, therefore to have no evolutionary future. Now ask what happens when you vaccinate a host. Well, the aim of the game when you vaccinate is to keep the host alive. That means that the kid gets this really nasty strain, previously would have died and removed the strain in so doing. Now the kid's better off because vaccination. But if that disease is transmitted on, that means a pathogen that would have been removed is now being transmitted in the community. So vaccination is enabling this superhot strain to persist in a way that it wouldn't have been able to do otherwise. Now that persisting is not such a big problem if it transmits on to another vaccinated person, because that person is going to be protected by the vaccine. But if it transmits on to a person who's unvaccinated, then the person who gets that is likely to die. In other words, vaccinating that kid puts another person at risk. So that was an argument that we made in the early part of the century. And the argument is essentially that vaccines are protecting these super hot strains from themselves, and so they allow these things to spread. Now, I know a bunch of you are probably pretty tense about cartoons, and me too. If it makes you feel better, here's a bunch of mathematics that went into our paper and a bunch of mathematics that other people did as well. And broadly speaking, what these mathematics show is that some vaccines could make diseases evolve to be more harmful. And we spent a lot of time trying to develop a model of malaria in a high transmission area based on Tanzania. We argued that some sorts of vaccines could create the situations where it became nastier. The key issue when I say some sorts of vaccines here is that there has to be onward transmission here. If the vaccine stops transmission so that the bugs don't go on to the next person, then there can't be any evolution. The evolution is only when you get what we call a leaky vaccine, where there's some transmission from an immunized person of wild-type pathogens to the next. If you don't have onward transmission, you don't have any evolution. If there's no onward transmission, then from the parasite's point of view, that host is an evolutionary dead end. Now, this was an extraordinarily controversial paper at the time, as you can imagine, especially amongst the vaccine development, the malaria vaccine development community. And I think the modelling efforts that have gone into this since our paper have upheld this conclusion, that in theory at least, or maybe a better way to put that is in silico at least, you can get the evolution of these hotter strains. But malaria vaccines, as many of you know, there's a candidate that's in stage 3 trials now, which isn't very good, and that's to say very effective. And I don't think there's going to be a malaria vaccine in my lifetime that is imposing strong enough selection for us to be able to tell whether this idea is correct. So I've, with one or two exceptions, slightly lost interest in the malaria vaccine area, and we've moved into looking at Marek's disease, which is a disease of poultry. Now, the first time I looked into a poultry barn, I have to say, as an infectious disease biologist, that's an exciting sight. 30,000 birds cram-packed into a small space. They're all the same age, same genotype, same immunological status, and if there's a virus gets loose in there, it's got 35 days to get across the flock before they go to the supermarket. So it's a very exciting evolutionary environment. And Marek's disease is a virus which is frequently found in these settings. It's an oncogenic herpes virus. It's transmitted by chicken dander, which is formed up at the base of the feathers and the feather follicles. So if you shake a chicken, the dust that falls out if it's infected contains virus, and birds breathe that in, and that's how the cycle goes. So from the point of view of studying it, it's very easy. When it was first identified or described in the 1920s, and then the virus itself in the 1950s, the virus didn't do that much. It caused paralysis in the birds, but it was transient and didn't otherwise affect the birds. Strains that do that, we now call mild Marek's disease virus. Since then, since the 60s and onwards to today, the virus has become progressively more virulent, and we now have strains called very virulent plus MDV. This is one of our experimental birds just before we put it down, infected with very virulent plus MDV. It's oncogenic, as I said, so this is, it can be very nasty. This is the heart of a chicken. One of the rights are covered in tumours that have been caused by this virus, the very virulent plus MDV. and it just destroys lymphoid organs. So it's hugely immunosuppressive. So you can see progressively more virulent strains there, really just totaling these lymphoid organs. Now, the really interesting thing to me is that this Marek's disease case is a case where vaccines did get undermined by changes in the virus. So the first generation vaccine was rolled out in the late 1960s, and that vaccine failed about 10 years later. Failure meaning here there were unacceptable flock losses, yield losses in flocks that were vaccinated. So there was a second generation vaccine rolled out, the bivalent vaccine, and that failed about 10 years later. And a third generation vaccine, Rispins, is the reason your chicken nuggets are cheap. So this vaccine is holding at the moment, and it is enabling the industry to operate, this and other vaccines against other diseases, enable the industry to operate in the way it does. So I think that story in itself is really interesting. It's like the drug picture. There had to be here a pipeline of vaccine discovery, even though it was only 2 more generations. There had to be improvements in the vaccines to cope with the failure, and that failure was due to virus evolution. And I should say that evolution has played out here as well as in Europe and in Australia, and in India it's playing out now. They're just moving to the Rispins vaccine now. There's no question that this is a result of virus evolution. This is the textbook in the area, and it's widely agreed that it's an evolutionary problem. Importantly here, there is no data whatsoever to suggest that this vaccine failure was anything to do with antigens or escape variants or any of that. There's no genetic changes in any of the known antigens. Antibodies that are elicited by the old strains are just as good at agglutinating the new strains. So there's no suggestion that antigenic variation or anything to do with escape variants or epitope changes or antibody escape, any of that has anything to do with what happened. This is why I got interested in it, because the only thing that's changed, the only virus phenotype that's changed over that time is that the virus has become more virulent. And this is one of those sort of semi-quantitative schematics that you get in nature where you can sort of see that a whole bunch of new symptoms have been added to the disease over that sort of time. I don't want to hang my hat too much on the exact quantitation of that, but nonetheless, it went from this thing that called mild transient disease to things that cause really severe, very, very severe disease and death. And is without doubt, I think everybody in the business agrees that something to do with intensifying the chicken industry, the commercialization of it and intensifying of it, led to increases in virulence. And the question that I got really interested in after our malaria paper was whether or not it was vaccination that drove this evolution. So we have a very large grant, or at least by evolutionary biology standards, to look at the evolution of this virus in the field settings and in experimentation. And this is a list of collaborators involved in this. I just want to mention Patty Dunn is the vet at Penn State who is the one that is able to open doors for us literally to get into the chicken sheds. And Venu Nair is my collaborator in Pirbright who is enables these experimentation. He's a viral pathogenesis person, and none of the work would be possible without him and these folk. So this is one of the farms we work on in Pennsylvania. That's Dave Kennedy, who's the postdoc who's leading the analysis of these data. We go into these barns, these houses, and pull chicken dust out and PCR it for virus, and we can get a sense of where the virus is and what's going on. This is just one of these farms, just to show you very briefly the sort of thing that's happening. We have 4 houses on this farm, so each house has about 30,000 birds in them. This is time along this axis. The grey bars are when the chickens have gone off to the supermarket and the barns are empty and cleaned out. New chicks come in. This is a new cohort. This is the point where the farmer changed to a different company and stopped doing one type of breed and went on to doing another type. So you can see, if you just look at this, that there are some houses, these are right next to each other on the same farm, and you can see that house 1 basically has no virus in it, nothing happening at all. House 3 gets a good epidemic here, a good epidemic here, something pretty persistent here. And then since the change in chickens, the virus is dying out. So there's quite a lot of heterogeneity in space and in time, and we're busy trying to piece that together. The only reason I'm mentioning any of that at the moment, because we're about a year into that surveillance program, is that what we have found is that, broadly speaking, the virus is in the broilers, the ones that we eat that go to the supermarket, not in the layer operations. So the layer operations are the ones that produce the eggs that we eat. That's the battery farming operations. And those ones don't have virus in them. The virus is in that setting. It's in the setting where the birds are going to the supermarket after just 35 days. So that's the sort of timeline we need to think about from the point of view of the evolution. So the experimentation that we've been doing is to ask, when we infect these birds with the hotter strains or the less virulent ones, what is the virus fitness like in immunised and non-immunised hosts? So we started this work, we were doing the work in isolators, and these are the isolators at Pirbright that Venu and his team work with. Chicks, we put chicks in there. The key thing from the point of view of the science I'm going to show is that the air is coming in these vents and out these vents. There are filters on these vents, and we can get the chicken dust from those filters, PCR those, and count the amount of virus that's in the air. These are the strains we've been working with and their virulence. So this B14 strain is the nicest strain in the world collection. This is a survival curve for chickens infected with B14 that are either vaccinated or unvaccinated. So this strain, B14, kills about half the birds that aren't vaccinated in 2 months. Okay? Now, when I say this is a nice strain, there is no human virus that kills 50% of us, at least not that's transmitted endemically. So this is an extremely lethal virus. Nonetheless, by Marek's disease standards, this is a very nice one. 571 kills most birds within 10 days, and then a bunch of them survive longer. 595, everything's dead in 3 weeks. MD5, everything's dead in 10 days. And now a few of the vaccinated birds are dying too. And then 675A, everything dead in 10 days. So that's 100% case fatality rate in 10 days. When I first saw that, I just couldn't believe that the virus was that lethal. Avian influenza is actually more lethal than that. It'll kill 100% of birds in 3 days. So these are extremely nasty viruses. And so the question we're asking, that's the strain variation that we've been working with, and we ask how would natural selection act on that sort of variation? When would it favour a strain that's this nasty, and where would it favour a strain that's that nice? How would natural selection sort those strains out? Were they present on the same farm? So with our incubator experiments then, we measure how much virus is coming off birds infected with those strains. And these different lines, each colour is a different one of the strains. And so the yellow line there is B14. All the birds continue to shed to the end of the experiment. The black line is the next nicest strain, and then these are the 3 hot strains that kill all the birds in 10 days. So in the unvaccinated birds, there's almost no virus shed before the birds are killed by those hot strains. So trivial amounts, whereas the birds that are living much longer with the nicer strains are shedding a lot of virus into the environment. This is what happens to the B14 strain if we vaccinate the birds. So the vaccination causes less shedding into the environment, so the virus titers are suppressed. A little bit less so for the next strain, but this is the key result. The vaccination keeps the birds alive, they've got these very hot strains, and so they now shed for the full course of that infection. Some of them are kept alive long enough that there's still virus being shed into the environment. And so if you put that together and ask how much virus in total is being shed from the birds, then we have the hot strain down on the left there and the B14, the nicest strain, on the right. Then you can see that in unvaccinated birds, there's a huge amount of difference in the total virus shed, so that the unvaccinated birds are shedding a huge amount of B14. They're shedding almost nothing of those hot strains because they're just not living long enough to shed. If you vaccinate the hosts, the story flips over. You get a 1,000-fold increase in the amount of virus being shed from the hot strains, and you get something less than that of a decrease in the amount of nice virus shed. So the fittest strains in an unvaccinated world are the nicest strains, and the fittest strains in the vaccinated world are the nastiest strains. But in this experimental setup, you can go one stage further and you can ask about what's actually happening with real transmission. So we do experiments where we put in 20 birds into the isolators, we infect half of the birds, and then we put them in with some sentinels and see what happens. And this is with 675A, the really nasty strain. In this experiment, all of the birds that we infected were killed or died by day 10, and none of the sentinels died. And we PCR the dust and we find almost no virus. So we know that's the reason the sentinels lived was that there was no virus in the air, they never got infected. If, on the other hand, we vaccinate the birds that we infect, we then keep them alive, and what happens is the sentinels die. And again, we know that's because the the infected birds are kept alive long enough by the vaccination to shed a bucket load of virus that can be picked up by the sentinels. It's extremely lethal and it kills them. So, to us, those data suggest that vaccination is making possible the persistence and spread of these superhot strains. In other words, vaccinating these birds killed these birds. Now, if we look at the three hot strains that we have there, these are the survival curves of the infected birds that we've experimentally infected. The unvaccinated ones in the dotted lines there, and they are dead around day 10, all of them. And then the vaccinated birds are in the solid lines. And that shows you how everything's dying rapidly. And then if you look at the survival of the sentinels, you see that the sentinels are all dying when you keep these birds alive with vaccination. The reason I put these data up is I just wanted to show you that the dotted green line, these birds, most of them are dead by day 10, but there's a few that are living to day 15. And that little extra bit of transmission is enough to cause a whole bunch of the sentinel birds to die. So just a little bit of increase in the transmission is enough to ensure the onward transmission of the virus. So it's very, very sensitive to how long those birds are kept alive. And the vaccination itself increases the lifespan of the birds and therefore the shedding period. So the vaccination is enhancing the transmission of these hot strains. Now, it's well known in Marek's disease and quite a few other diseases that there's maternal protection so that the mothers can pass on immunity to their offspring, and that's no different here in Marek's. And so you can vaccinate the mothers, and that protects the offspring. So we did the same sorts of experiments with these hot strains where this is just a repeat of the controls. If you experimentally infect the birds when they have no protection, they all die and the sentinels all survive. If we then vaccinate the mothers and then take the chicks of those vaccinated mothers, then they do die, but they stayed alive now. So these guys are all dead by day 11. These started dying at day 11, and so there was another 20 days or so where these were alive, and because of the maternal protection. And of course they're shedding virus during that time, and so we can measure that, and then we can see the sentinels start to die, and they're all dead by day 32. So vaccinating the mothers also allows these hot strains to transmit in the environment. We did the initial experiments with Rhode Island Reds, which is a sort of representative strain of what a chicken might have been like in the 1970s. These days chickens are high-performance machines, and various reviewers suggested that our data wouldn't hold up if we did the work in a modern and commercial setting where you use these fantastically fast-breeding birds and the new genetics, maternal antibodies, and vaccines and so forth. So these are experiments which we've been just finished in the last few months. The European Union regulations for chicken experimentation has forced us to give up on the isolators, and we now do the experiments on floor pens. It is amazing to me that there's billions of chickens in Europe that are kept in those farming conditions, and yet the experiments with our 20 or 30 birds are really tightly regulated. Madness. However, it has led us to do these experiments on the floor now, so it's very close to a real barn setting, real farm setting. But we do the same sorts of experiments that we were doing, and so now what we've got is birds that we're buying commercially, so they're vaccinated, they're maternal, they themselves are vaccinated, their mothers are vaccinated, and then we've got sentinels which are commercially, we buy them from commercial producers, so that means their mothers were vaccinated, and we vaccinate half of them or not. And again, what happens now is that the sentinels all become infected, so our birds, our donor birds if you like, are all shedding virus, and our sentinel birds become infected. And that means that modern birds are not refractory, so the modern birds are shedding and modern birds are getting infected. There's nothing funny about all that. And the sentinels, well, depending on whether they're vaccinated a lot, they die at certain, fast or slow, they do die. So the same picture is holding up in the commercial birds, that if you vaccinate the birds, that allows the virus to transmit and infect new commercial birds. You can also look at the sentinel birds and ask, how long do they themselves shed for? So this is the shedding periods from the birds that have acquired the infections from the experimentals, and this is the period of time they're shedding for. The crosses is where the birds died, and so you can see that and this is, we did the experiment twice, you can see that the unvaccinated birds die earlier, and so vaccinating the sentinels has also kept them alive longer to shed for longer. So the next generation of transmission is also enhanced by vaccination. So putting all that together then, that suggests that vaccines keep the birds alive long enough to shed properly, shed these hot strains into the environment. Vaccinating the mother does the same thing, and the situation is not changed in commercial birds. So to my eyes, what this is showing is that vaccination is enabling these strains, which are super hot, to persist in the world that they wouldn't have been able to persist in before. And so then you could ask the question of whether or not vaccination has made Marex so virulent. I'm a little bit unclear about that, or unsure about that. I think what our data show is that vaccination is sufficient to explain the persistence of these lines, these hot strains. So whatever caused them to evolve, they can persist now because the chicken world is vaccinated. It might be that the vaccine itself drove everything and was the cause of the evolution in the first place, or it might be that other things like the shortening lifespans of the birds, the high density situations, all those things will contribute to the evolution. But whatever caused it or drove it, these days, the vaccination is enough to allow these super hot strains to persist. So that's my third point, that vaccination can, under some situations, make possible the strains of pathogen strains that are otherwise too lethal to persist. Okay, the fourth thing I want to talk about is a question which is naturally raised by that. Is there something special about Marek's disease? Is there something special about chickens? Or could this happen in other contexts? I believe it can. I mentioned before the feline calicivirus story. That's a case where I think the strains that popped up were so hot they could only persist in vaccinated cats. And even that, they were so hot, once they ran out of, the cats were dying even at such rates, it was hard to maintain those viruses except in the high density situations where the vaccines were being used. You may or may not buy this analogy. Myxoma virus was used for biocontrol against Australian rabbits, a textbook example of evolution of virulence. We've been trying to put the genetics into this story, and I don't have time to go through that, except to say that strains of the virus, so when they released the virus in the beginning of the 1950s, the virus killed 99% of the rabbits that was in Australia. It then evolved less virulence through time, because the lethality was so high that there was a transmission advantage to lower virulent strains. The rabbits then started evolving resistance, and as the rabbits evolved resistance, the virus became more virulent again. We know that because the Australians did very, very excellent experiments with lab rabbits and lab strains of virus. We can see that the resistance has gone up in the rabbits and the virulence has come up again in the virus. We've been phenotyping modern strains of the virus in Penn State. These are the rabbit experiments going on. It's Peter Kerr, who's the key collaborator here in Australia, the last myxomatosis man in Australia surviving, as far as I can tell. We've been taking lines that he's been collecting from the field now and putting them into rabbits. Those strains are now hotter than the original ones that were released. So the original ones that were released had a 99.9% case fatality rate. These strains have a 99.9% case fatality rate, but they kill them even quicker. And they kill them before they've even developed proper symptoms of myxomatosis. The virus has become extraordinarily hot as measured by putting it into a lab rabbit. Now you don't see that in the field because the rabbits have become more resistant. So the resistant rabbits have created a virus which is hotter than anything that's been seen before in myxomatosis. When it first happened, it was horrifying. It was carnage in the animal house. They bleed out. So they bleed into ears or legs. It's just very, very scary. And they drop dead very fast. They go from being okay to massive bleeds to dead in a few hours. It's really incredible. This virus has become very, very hot. And what's happened is the rabbits have become more resistant. So coevolution led to an increasingly resistant rabbit. And I think that's the same as what happens with vaccination. that you protect the the animal either by genetic enhancement or vaccination or coevolution generates resistance or whatever. As the host becomes more resistant, then the pathogens can become more virulent. Nonetheless, there are many people who keep saying to us there is something special about chickens, and it is true that chickens are pretty special, and obviously they aren't people. But it is tempting to ask, what's happening in the human situation. And I'm going to just spend 2 minutes on the difficulty of knowing what's going on here. These are data from a paper on diphtheria from India. This is the second half of last century. And the bars are the number of cases in a hospital of diphtheria. And you can see that after vaccination came in, the number of cases of diphtheria in that hospital dropped dramatically. But the line there, the dotted line, is the chances of dying of the disease if you get it. And so it starts out at 10%, and then after 50 years, you've got a 30% chance of dying of diphtheria in that hospital. Now, it's tempting to say, well, look, there's the virulence has gone up over that period coincident with the vaccination. And that might be consistent with what I've just told you. It might not be, though, and the authors of this paper suggest that it's the funding in their hospital. So they got less and less resources after the British Empire fell apart, and so their hospital got less good at dealing with diphtheria. So that's what they attribute that rising case fatality rate to. And I can't assess that. For all I know, that's a perfectly good explanation. The only way to actually tell whether that's evolution is to take the strains from 1950 and the strains from now, stick them into people, and see which dies fastest. And of course, that's exactly what we've been doing with the chickens and the rabbits. That's why we can tell what's happening with virulence, because we can do those experiments. With the human case, we can't, for obvious reasons. And so we're stuck with animal models. And this is what happens when you're stuck with animal models. You just don't know what's going on. So this is the paper we had. You do know what's going on with the animal models. You don't know whether it's relevant to humans. And so we have in the paper our big caveat section, which is going on about the fact that we recognize that mice aren't humans. Referees really do ask me to put that into a paper, as if I didn't notice. What I think our data, and so the referees also asked us to put in another box, evaluating risk, where we restated all the caveats. You just don't know, right? And when this paper came out, we got quite a lot of journalists were interested, and the way I kept putting it to them is I think our experiments show, the mice experiments and the chicken experiments, show that this possibility that vaccination could drive more virulent strains, the experiments show that the probability of that happening is not zero. That under some biological conditions, in the chicken houses and in our mice in the lab, this evolution can happen. So the probability is not zero. It's somewhere between a little bit bigger than 0 and 1. And we just don't know. But you've got to ask the question, with these diseases that we're working on now or vaccinating against now, the new ones, as well as all of the veterinary situations, what do we think is happening? What's going on in these situations? What could be going on? And I think the way to think about this is to ask, or to try to assess the risk, is to ask, where are we going to see this onward transmission? And so I think when we've got vaccines that are starting to, which are not preventing transmission, which are targeting subsets of strains, or which are targeting virulence determinants, or which can be overcome by immunosuppressive substances, these sorts of things where you can get onward transmission, these are the sorts of vaccines where we have to start asking, will they drive virulence evolution? So broadly speaking, all of the things that would allow ongoing evolution are the sort of things that we have to get a bit nervous about. And then for a risk assessment to say, well, would we get the evolution of increased virulence? I think the question is, the way to handle this, to think about it is to say, are super hot strains possible? So is there, in gene space, if you like, the possibility of mutations to hyperpathogenicity? What stops super hot strains spreading in the pre-vaccine era? Why aren't our pathogens nastier now? Ask what causes the pathogen virulence to be kept under control, and then ask if the vaccination is going to relax that. If vaccination is going to take something that's stopping hot strain spreading and make it weaker, then we have to get concerned. And then we can do experiments, of course, at least again with the caveats involved with the animal models, and say, are we likely to have strains which have a fitness advantage in vaccinated hosts? And that actually you could do to some extent in clinical trials and in following pathogens after clinical trial and vaccine rollout. So my fourth point is that this issue of evolutionary risk, I think, is real, but assessing how important it is really tricky. And maybe we're lucky. Maybe it is something that applies in some contexts like farming and isn't going to apply in the human case, in which case it's still a significant issue for industry, but might not be a human health case. Where we're taking this work now is asking in the Marek's case whether the latest generation vaccine is likely to fail. We're trying to do molecular epidemiology on the farms to figure out where the strains are spreading. We're looking for what we're now calling hyperpathogenic strains, so ones that are even more lethal than the ones we're working with. We have various ideas about how you could change farm management to alleviate some of this evolution. We want to work on genetically modified chickens to see if they'll do the same thing. If you enhance the resistance of the host, either by GM or by selective breeding or by vaccination, it should have the same effect. And we can ask questions about the evolution of the virus at the genomic level. And then in terms of other viruses, other diseases rather, we're interested in several other poultry viruses, the fish farming, the rabbits I mentioned. We're doing some work on liver stage malaria vaccines, and I continue to be very interested in monitoring what's being published on the HPV, pertussis, rotavirus, and diphtheria story. So these are my 4 take-home messages, and I think I'll leave it there. Thanks very much.
Okay, what I'd like to do is we'll take 5 minutes for questions from the general group. We'll take a 2-minute break, and then those who would like to leave can leave, and those who would like to stay for further questions can stay on. So, Andrew, I'll let you do the question.
Okay. Oh.
And we need this because it can take.
Thanks. So a lot of the data, could you comment on the size of the genome and ability to actually make hypervirulent? Because a lot of the data with the smaller viruses that when you have a vaccine, you actually have a fitness cost to the pathogen rather than selection of hypervirulence.
Yeah, so it may be no accident that Marek's disease has got getting up to 200 genes and myxomatosis is up to 200 genes. It might be that it's no accident that we're working on large viruses that might give them more, as it were, things to play with from an evolutionary perspective, fewer constraints. In terms of the fitness costs of these mutated strains, yeah, I mean, I don't think we really have very good estimates of that. The fitness cost that we measure when the chickens, there's the killing the host, is an overwhelming issue that's going to swamp out any minor differences in viral growth rates and those sorts of things, I think.
So the second question has to do with that then, because a lot of times the pathogenicity is due to the immune response to the pathogen rather than the pathogen itself.
Yeah, that's right. So if I'm going to formally define virulence, and what I mean by that here, I mean those aspects of survival and morbidity that are encoded in pathogen genes. And that can include genes which encode products which interact with immunity and cause the virulence. But I'm really thinking, and that's often the case, but I'm really thinking here about the evolution of the genes that are involved in that side of things. Yeah, if it's entirely host-driven and there's no pathogen variation in virulence determinants, then the evolution I'm talking about is not going to happen. You need genetic variation in the pathogen in order to get pathogen variation. And it could be that the host variation swamps that out, for sure. The back.
So, do I have to do this thing? So, one of the things you didn't mention in your comparison between antibiotics and vaccines is antibiotics are given therapeutically, almost exclusively, and vaccines are almost exclusively given prophylactically. And so, where you've pre-armed the immune system to a relatively small number versus wait till you have symptoms, you have a very large number. And do you want to comment on that? That certainly is an important aspect of it.
Yes, that's right. If you ask the question of why is it that some of these childhood diseases are so apparently evolution-proof, one of the answers you might give to that is along those lines, that the pathogen populations that are trying to get into an immunized host are very small, and so they themselves don't have much mutable potential because of the smaller population size. There are other sorts of explanations as well. That's, I think, a possibility. Another one is that drugs typically target rather fewer elements of the pathogen. They typically, after one enzyme, for example, whereas immunization is typically targeting many different antigens, not always, but often. So there are a bunch of those sorts of explanations. I agree. I guess my point was really that it is an interesting thing to say why drugs seem to be different from vaccines, and I guess what I just wanted to emphasize is that among the vaccines and the diseases, there are different types. And, the childhood diseases are, I think, very special cases. Charlie.
Andrew, first of all, I get nervous about what the press thinks about that vaccines make things worse.
Yeah.
Because we live in a society where there's a segment that wants to believe that. So just making that statement, then going back and looking at your two...
Could I just, because you're probably going to ask a different question now, right? Can I
Yeah.
just comment on that? I do too. Vaccines are one of the most successful public health measures we've ever had. We shouldn't, all of us, should do our very best to make sure everybody vaccinates themselves and their kids with everything we've got. Not making an argument against that at all. In fact, if anything, my argument is another reason to get yourself vaccinated, because although you might drive the evolution, you're protected from it. So,
Well, that's a very self-serving
self-serving,
point of view,
but I can also just make one other comment. I think we are in a difficult issue here where we are trying to investigate the science of public health that has public health implications, and that's true of our drug work as well. Right. However, I do think we need to be aware and very conscious of public perception. I do not think that should stop the science.
Oh, I agree with you entirely.
So we have to be super
But let me
careful
then, with that point, let me try to evaluate your two systems. You've got a chicken disease, you've got a rabbit disease. The time of variation of your pathogen really kicked off in the 40s. Now, I don't know if that's just because of the experimental tool, but I'm reminded that's when commercial chickens became available to farmers. It took off dramatically in the 60s. That's when hybrid chickens became available. Essentially what farmers were doing with their 30,000 chickens in one shed was greatly narrowing the genetic variability in the host.
Yes.
And thus making it, so I would argue that the vaccine is really a tool to study the genetics of immune response to pathogens, and you've provided the experimental system by narrowing the gene pool. And it wasn't the vaccine that drove it, it's the cultural utilization of a narrow germplasm. Same thing happened in Australia, where you knocked out almost eradicated the rabbits. What you're left with is a gene pool that had the capability then of allowing this virus to persist. So I would, if I were explaining your data, I would be saying vaccines are a tool to study the genetics of immune responses or host pathogen interactions.
Well, that is true, but I'd make a stronger claim than that as well. I think what our data is showing is that minimally showing is that the host genetic diversity that you see under current conditions, coupled with the vaccination or the resistance enhancement through the rabbits, is enough to maintain these hyperpathogenic strains. If the genetic homogeneity in those systems enables that vaccine-driven evolution to proceed, that's good news, because then it won't be a problem for us. I mean, for people, it's bad news for farmers and aquaculture and all of that, because they'll have persistent, ongoing problems with these things. I think we can actually make some headway with that problem, with that assertion of yours. We can ask, because if the genetic homogeneity is what's allowing this to happen, the flip side of that is the genetic heterogeneity stops it. So that diversity means that a strain that's super hot in one genotype of host will be a cold virus in another. That would, if that was true, that would cause some significant natural selection would not be seeing the viruses in the same way. You with me? Okay. So when we take our hot strains and our cold strains, the hot strains are still hotter in any breed they've been tried in. Now, there are more or less resistant breeds, but the rank order of those strains stays the same. So the hot strain is always hotter and the cold strain is always colder, no matter what genotype it's in of chicken. So selection is still seeing the rank orders of those, no matter what the genotype is. Now, I grant you that having a genetically homogeneous population is going to make virus adaptation easier in a sense, but in fact, the trade-offs that you would need to see between one genotype and another to slow or stop or even reverse that evolution, there's not evidence of that. There's no question the host genetics matter, but there's no question that there's no evidence that there's a strong virus by host genotype interaction. So in other words, that the hot strain is not hot in one host and cold in another. So natural selection is always seeing the hot strain as hot.
Let's take one more question in this session from Roy. We'll take a 1-2 minute break so folks who have appointments can leave, and then more questions can happen. So Roy?
I want to switch to and get your opinion on another aspect of the evolution or the de-evolution of pathogens. And you kind of alluded to the fact that it's in the best interest of the pathogen not necessarily to kill the host. And as I well know, Salmonella separated from E. coli 100 million years ago and killed all the lizards and amphibians. They would have driven Jim Collins crazy if he was around then. But now Salmonella is innocuous, and he's got other critters that are the problem. But and by the way, we can talk about this later. But Salmonella killed the dinosaurs.
Okay, I'm agnostic on that.
Response.
Yeah.
But you know, and so Salmonella now is a commensal in all amphibians, reptiles, most birds, with the exception of two diseases, and now is commensal in many mammalian species. And so the bug is figured out, you know, in its best interest, yes, to persist and be transmitted, but not to cause overt disease. And that seems to be a fairly significant selection against virulence, I guess, from the point of view of your definition of virulence. So,
Yeah, I mean, I think, so your question really is about the variation in virulence that we see among different pathogens, or we're going to call them commensal, different organisms that infect us.
but over billions of years of time. So.
Yeah, no question. I mean, I think there are cases where long-term coevolution leads to avirulence. I think there are other cases where long-term coevolution hasn't led to avirulence. So the problem for an evolutionary biologist is to explain why those different trajectories happen. And the sort of explanation I would give is in terms of the kind of ways, things that I've been talking about. How long are the infectious periods? What's the cost of killing the host? And so on. So one presumes in the case of Salmonella that hotter strains of Salmonella have a fitness disadvantage compared to these symbiotic ones, either in terms of the length of the infectious period or the number of infectious stages that are produced. And that sort of explanation would explain your trajectory. The trajectory on farms for many of these viruses is going in the other direction.
All right. So what I'd like to do then is first